In this video, I am going to talk about the transport of oxygen and carbon dioxide in the body fluid. First and foremost, when oxygen is diffused into the alveoli, it will enter the bloodstream and transported to the tissue, while carbon dioxide will diffuse out of the blood and into the alveoli to be expelled from the body. Based on the oxygen dissociation curve, partial pressure of oxygen increases when the oxygen binds more to the hemoglobin. However, the affinity of the hemoglobin for oxygen may shift to the left or right depending on the environmental condition. As the partial pressure of oxygen increases, the hemoglobin increasingly saturated with the oxygen. There are only 1.5% of oxygen in blood that dissolves directly into the blood itself, while 98% bound to protein, called hemoglobin, and carried to the tissue. There are three methods of transportation of carbon dioxide in the blood. First method is dissolution directly into blood. Dissolution directly into blood, there are only 5 to 7% of the carbon dioxide that dissolves into the plasma. Second method is binding to the hemoglobin. These methods only have 10% of carbon dioxide that bind to hemoglobin, while the third method is carbon dioxide is carried as a bicarbonate ion or act as a bicarbonate buffer system where there are 85% of carbon dioxide that diffuse into the red blood cell.
Next, I would like to present about a question. Does the temperature affect the carbon dioxide and oxygen diffusion into the body fluids? So basically, temperature will affect the diffusion by reducing the hemoglobin affinity towards the oxygen. For example, when one to exercise, the muscle tissue will increase the temperature by about 37 degrees Celsius, so that the higher the temperature, the easier the unloading of oxygen. So as you can see, referring to the figure 1, it's shown that the curve is shifted to the right because of at the temperature of 45 degrees Celsius. You can see that the oxygen association with the hemoglobin is lacking. As well as to carbon dioxide diffusion, when one tissue is in a high temperature, it will experience the lag dissociation of carbon dioxide from the hemoglobin. This is because of the carbon dioxide is following the Henry's laws, which is it defined that the number of molecules in solution is proportional to the partial pressure to the liquid surface. And according it, the carbon dioxide is giving a value of 0.0308 mol per liter at 37 degrees Celsius, so that the association of carbon dioxide in the body fluids is getting higher when the temperature is high. So in simplicity, both the diffusion and solubility, the higher of both these characteristics of carbon dioxide will ease the diffusion in body fluid when in at high temperature.
So for the next question is inorganic ion affecting the respiratory pigments? Inorganic ion can act as a modulator of respiratory pigments. The concentration of specific inorganic ion in blood cells sometimes allosterically modulates the oxygen affinity or other attributes of respiratory pigment. For example, the concentration of chlorine in red blood cell is critical allosteric modulator of hemoglobin function in many ruminant mammals and bears. Other than that, the bicarbonate ion is a modulator in crocodilians. The last example is the divalent ion, which is calcium and magnesium, are important allosteric modulators of hemocyanin in crustaceans.
So I would like to talk about the respiratory pigment. It's a molecule which increases oxygen carrying capacity of blood. Generally, there are four most common respiratory pigments, which are the hemoglobin, hemocyanin, hematine, and chorocortin. So the first one, hemoglobin, is an iron-containing oxygen transport metaprotein in red blood cells of most vertebrate and tissue of some invertebrate. It is made out of four subunits, which are two alpha units and two beta subunits. It increases total blood oxygen capacity 70-fold compared to dissolved oxygen in blood. Besides, it carries oxygen from lungs or gills to the rest of the body, and it also transports carbon dioxide as carbaminohemoglobin and nitric oxide bound to a globin protein PO group. It is bright red in color when oxygenated and dark red in color when deoxygenated. Hemoglobin is present in mammals, fungi, and plants. So next is a hemocyanin. It's a protein which transports oxygen throughout the bodies of some invertebrates. It generally consists of two copper atoms that reversibly bind a single oxygen molecule and is found suspended directly in hemolymph. It's blue in color when oxygenated and colorless when deoxygenated. Hemocyanin is found only in mollusca and arthropoda. So we continue to hemetrine. It is an oligometric protein responsible for oxygen transport, and it exists as a homoattamer or heteroattamer composed of alpha and beta type subunit of 13 to 14 kilodalton, and is generally found in cells or corpuscles in blood. It is 1/4 as efficient as hemoglobin, and it is violet pink in color when oxygenated and colorless in color when deoxygenated. It is found present in marine invertebrate phyla of cipoculids, priapulids, arthropod, and single annelid worm genus Megalona. So the last pigment will be the chorochorin. It is an iron profiling pigment and oxygen binding hemoprotein. It is found free floating in plasma blood and is dark green in color when oxygenated and light green in color when deoxygenated. Generally is present in most annelid and particularly certain marine polycysts.
So let's move to the next question. It is an interesting question regarding respiratory pigment in animals, which is how blood can become green. Does animals like human has a time when the blood will becoming green? I will explain it later. So basically all animals which is vertebrate has red blood color except for a type of lizard which is green-blooded skink. The green-blooded skink possess lime green blood, green tissues, green bones, tongues and mouth lining. Actually, something had happened in circulatory system that causing them to have lime green blood and other physical characteristics which is in green. They have red blood cell full with hemoglobin which is red in color. But generally in animals, the red blood cells have short lifetimes which will be taken out from the circulation by the liver. When the cells are trashed, the hemoglobin will be recycled. This situation is very different in humans and skink. In human, the hemoglobin recycled to biliverdin, a green chemical. Then the biliverdin will be rapidly converted into bilirubin. Bilirubin is a toxic compound, so the liver filters it from the blood as quickly as possible. If the bilirubin builds up to the high level, the skin becomes yellow. Hence, a condition called jaundice can happen and can be fatal. But in skink, the hemoglobin disposal is stopped halfway. The skink can't convert biliverdin to bilirubin. Hence, the biliverdin builds up in the blood and the green color is over the red color of the blood, even though the respiratory pigment is red. So, for the result, the amount of biliverdin is 40 times higher than the highest level ever measured in human being.